Thank you for joining today's session in multimodal preclinical imaging titled Targeting Contrast Agents. My name is Marty Pagel and I'm at the MD. I'd like to declare conflicts of interest, some companies I work with, some collaborations and funding sources. So I'd like to begin this topic of targeting contrast agents by first discussing targeting. Typically, we think of an antibody as a targeting moiety that's labeled for MRI and that can identify an epitope which is on a cell receptor. Targeting sensitivity is really dependent on the number of receptors on the cell, which can range from 10 picomolar to 10 micromolar. MRI sensitivity in a voxel is approximately 10 micromolar or higher, depending on the quality of your instrument and the contrast agent. So you can see right away that it is challenging to detect cell receptors that are below the detection threshold of MRI in most cases. We'll discuss some alternatives to this. Also, I think it's important to recognize that there's a difference between target specificity and target selectivity. Specificity pertains to the antibody itself. If the agent, the antibody, binds to one epitope on the cell receptor, and here you can see one epitope that fits within the binding pocket of the antibody, then the antibody is said to have good specificity for that cell receptor. Selectivity is a term that pertains to the epitope itself on the receptor. So if the epitope is unique only to that receptor, then the antibody is said to have good selectivity for that one and only one receptor. However, if this epitope, this binding motif on the receptor, is also present on other receptors, well, the antibody has good specificity for the epitope, but does not have good selectivity for the receptor because there could be multiple receptors with this epitope. So selectivity deals with the epitope, and those two terms often are confused, and I just wanted to clarify. There are two types of targeting explored by MRI contrast agents. One is active targeting, typically limited to the cell surface and extracellular tumor microenvironment, because getting enough MRI contrast agents inside a cell and still having the cell be viable can be challenging. And these active targeting motifs are typically antibodies, but could also be peptides, and some other targeting systems have worked as well. I should clarify that I work in a cancer center, so if I say the word tumor cell rather than just cell, I apologize in advance. It's just human nature for me to do that. Yeah, passive targeting systems, though, can be advantageous as well, and they don't target a specific molecule within the tumor microenvironment or the cell receptor, but target a macroscopic characteristic of the tissue itself, which then, of course, depends on the macromolecular composition within that tissue. Glycosaminoglycans in cartilage are an excellent example. I'd like to show that on this next slide where the assessment of cartilage deterioration can be interrogated by using a contrast agent in a passive way. So the contrast agent is negatively charged and will then perfuse into the tissue, especially in regions that have low GAG content because the glycosaminoglycans have a negative charge. In high GAG content areas with a lot of negative charge, well, the negatively charged contrast agent will not accumulate. And so you can see in this schematic, a lot of agent did accumulate in this low GAG environment. And that can be seen in an MR image of a piece of cartilage that had low GAG content where the contrast agent was detected using T1 weighted MRI. And that was confirmed with ex vivo staining where the GAG content was low in that portion of cartilage. And this is really an excellent example from Martha Gray and Jim Hartke from many years ago in the early 2000s. And still, I think a great example of this method known as DGEMRIC or delayed gadolinium enhanced MRI of cartilage. So passive targeting technique. But of course, we like to think of active targeting. Here is, I think, the one of the first examples ever published. There are a couple right around 1999 and 2000 that appeared back to back. So one of the pioneering studies performed by John Kodak and Stacia Anderson and colleagues, where they had a rather complex system with F19 and gadolinium DTPA for MR imaging via fluorine or T1 weighting attached to a biotin evidence system for ease of labeling to an antibody that targeted alpha V beta 3 on the vascular endothelial cell. And so this experiment was performed by implanting a growth pellet within the eyeball of a rabbit. And of course, this was done under all humane conditions. The rabbit was under anesthetic during the imaging, and an MRI coil was used to pick up the signal of the contrast agent that would accumulate, where the pellet then stimulated the expression of the alpha V beta 3 receptor. And here's a schematic of the imaging plane through the eye and the place where the pellet was injected. And of course, the pellet was injected days earlier in order to stimulate the alpha V beta 3 expression. Pre-contrast, nothing remarkable. Post-contrast, there was up to a 17% signal enhancement due to the accumulation of the agent, presumably due to the presence of the receptor. But you can see this as a mild increase, 17%, and a pellet had to be implanted in the eye. So there is a very large expression of the cell receptor, and still just barely enough signal enhancement to see it in this image, which brings up an important point. That, it takes a lot of gadolinium to detect these cell receptors. Can we get enough gadolinium to the site of the cell receptor? Well, let's look at a worst case example where cell diameter is 2.5 microns. Best case example where cells are very small, so there would be a lot of cell membrane and therefore a lot of cell receptor. And what happens if the intracellular space is low relative to high? Well, we can add that to our criteria. As a side note, tumors often have low intracellular space. They're growing very rapidly and a lot of extracellular space. So this would be, unfortunately, a worst case scenario for tumors. The number of receptors per cell is really the overriding factor that determines if MRI contrast agents based on gadolinium or iron oxide can detect a cell receptor. And so in the best case scenario, you could have a lot of overexpression of those cell receptors. Number of cells that actually have the receptors is never 100% within the tissue, but let's look at a low worst case to best case example there. And then finally, the voxel, at least preclinically, in a small animal MRI scanner is typically 90 nanoliters, 300 by 300 by 1,000 micron. And you can then calculate, well, based upon the volume of a cell and all this other information, what's the receptor concentration within that voxel, that 90 nanoliter voxel? And so here are the three numbers, 10 picomolar to 20 micromolar. Again, showing that range of receptor concentrations that I showed earlier. Well, for gadolinium DOTA, now with a typical R1 relaxivity of 4 per millimolar per second, while the minimum detection level is approximately 50 micromolar, depending on the quality of your MR scanner. And so you can see that worst case scenario, you'll just never be able to detect that cell receptor. Even on the best cases, you would need 3 gadoliniums to bind to that cell receptor. And so you'd have to design something a little clever. If you attach this to an antibody, well, you can get an enhancement factor, let's say a factor of 10, if you put 10 gadoliniums per antibody and still retain the antibody integrity. But in an average case, you're still not able to get enough sensitivity. And in fact, even when you go to a nanoparticle system that might actually increase the sensitivity by tenfold again, well, you never really can reach an average case using a gadolinium-based MRI contrast agent. Might have a better chance with an iron oxide particle, which has a little bit better sensitivity. But I think you get the idea that it's very challenging to detect cell receptors because the receptor density just isn't there enough for detection with MRI. However, there are alternatives. We could detect a very abundant protein within the extracellular tumor microenvironment, not a cell receptor. In this case, collagen, studied by Peter Caravan and colleagues, was targeted with a collagen 1 targeting peptide that was labeled with gadolinium. You can see uptake after almost an hour, showing that there was an enhanced uptake and retention of the agent in this tumor. In the control tumor that did not have high collagen content, the agent basically washed in and washed out of the tumor as normal. And you can see the difference between I'm sorry, this is a control agent on the same tumor type. So the targeting agent worked well on the tumor that had a lot of collagen. The control agent did not, showing that there is some detection of collagen in this case. Z.R. Liu and his colleagues have been detecting fibronectin. Here's a great case with a tumor model with high, medium, and low fibronectin. Fibronectin targeting in the high fibronectin tumor model was successfully accomplished relative to a control agent that washed in and out of the tumor much faster or at a lower amount. The medium and low tumor models that had less fibronectin showed less uptake. And that can be seen in these histograms showing that over the course of 40 minutes, the tumor model that had high fibronectin did indeed have a higher contrast to noise when the fibronectin detecting agent was injected. So, nice complementary study showing that different tumor models with different amounts of fibronectin can be distinguished. And in this case here, the targeting agent versus a non-targeting agent showed nice difference for the collagen study. But again, the difference between targeting and non-targeting is rather small. And so these studies are challenging to perform because you have to get the timing right, the amount of agent has to be set correctly, and of course you need a model that has high fibronectin and low fibronectin so you can compare the two. If you only had the low fibronectin model and you saw these white bars, well, it would be more challenging to know if you're detecting fibronectin or not. This is a differential measurement between the tumor models. So Guanshu Liu performed a remarkable study using chemical exchange saturation transfer, or CEST MRI, to detect PSMA, a very abundant cell receptor on some types of cancers. In this case, glucose has exchangeable hydrogen atoms that can be saturated so that their MR signal disappears. So before saturation, you can see it within this CEST spectrum. Afterwards, MR spectrum. And in this case, you can't see the signal here in the spectrum. Um, After chemical exchange of these saturated protons with protons on water, you can see the blue balls have now changed to red. Well, a lot of the water hydrogen atoms are now saturated, and so the total water signal decreases. So that's the CEST effect. Well, what Dr. Liu did was to take the glucose and really have a polymer, a dextran, of multiple glucose monomers. So he had a very high number of hydrogen atoms on these hydroxyl groups, and he was able to pick up the CEST effect as as a PSMA targeting moiety labeled with the dextran, perfused into the tumor, and then was retained relative to a tumor model that did not have PSMA expressed. So you can see a difference between the targeting of PSMA and not targeting of PSMA in the other tumor model. And of course, with dextran alone, there was no particular uptake. You needed the urea-based targeting moiety to detect the PSMA. So really remarkable that a very insensitive MR technique, CEST MRI, can actually detect cell receptors if the system is designed in a clever way to have a very large amplification factor, and that is the key. In fact, here's another example of amplification using, in this case, an iron oxide nanoparticle to detect HER2, a great study by Ding et al. That was published in Molecular Imaging and Biology. This schematic is from a great paper by Patricia Rivera Piera, really a PET radiochemist, a PET applications person. But a really nice paper that shows the targeting of HER2 with PET, and I just borrowed the figure here for didactic purposes. But if you have an agent that binds to HER2, that becomes internalized into an endosome, and then either lysosomal degradation occurs and the agent is released, or the HER2 can recycle back to the surface, also allowing the contrast agent to be deposited into the cytosol and accumulate in the cytosol. So this recycling mechanism is an amplification process that allows a lot of these gold stars to be accumulated in the cell and get high contrast. And that can be seen before and after injecting the agent, 24 hours after in this case. The blocking study showed that there was no accumulation of the agent when the HER2 receptor was no longer available due to blocking. And that can be seen in this chart over here as well. So really a great example that if you have amplification, that will greatly help to detect tumor cell receptors. But you have to be clever in how you choose your biological system in order to get this amplification. One other topic I wanted to bring up about binding. This is more from fluorescence imaging, but can apply to MRI as well. You get improved specificity if you try to detect two cell receptors at the same time with a linked system, two different hands, so to speak, that are binding to the two receptors. However, if one of these hands is weak, so much that the binding potential between the yellow and green hands here differ by greater than 100, well, really, this green hand really doesn't matter, and as if you have a monovalent system. So the binding potential is the binding affinity times the receptor concentration. So not only is it the affinity for one of these binding moieties to the receptor, but it's how many receptors are available too. So if you have poor expression of one of the two receptors, well, you're back to just a monomeric system, and so you lose the improvement from heterobivalency. So heterobivalent systems are intriguing in vitro, have been very challenging to get to work in vivo because you have to have a balanced system with two moieties that have the same, or roughly the same binding potential. Hasn't really worked out that great. I'd like to point out that there is a myth of the size threshold. We can label a variety of nanoparticles, antibodies of different sizes. They're all listed down here. And really, we want our imaging agent to wash in and wash out of the tissue relatively quickly, at least within a day. That's different than chemotherapeutics, and even some nanocarriers with intermediate sizes will last in the body for days as well. And so very often I hear, oh, if you have a certain size, you'll be okay for imaging, or if you're at a certain size, that's fine for chemotherapeutics. But I really think you have to consider the delivery time and the amount. We want a large amount of MRI contrast agent in a very short time, and that's very different than a chemotherapeutic, where we want a small amount relatively to be delivered, but over a much longer time. And so you have to consider the whole pharmacokinetics of the system. Also, a very nice paper by Brian Pogue and his colleagues, Scott Davis and others, was published in 2013. Just pulled out the numbers and calculated that. And it appears that the difference between the targeting and non-targeting agent is maximized at 2.8 hours. Once again, showing that at about 3 to 6 hours for a lot of these larger systems, larger than a small molecule, um, that really is helpful timing for determining the difference between targeting and non-targeting. All right, well, that concludes targeting agents, but I did want to point out a few aspects of responsive agents for comparison. And really, a large amount of work has been done over the last 20 years now. I've only done the analysis over the first 15 years of this century to detect enzymes, pH, ions. Well, pH is also an ion, and then other biomarkers as well, as you can see here. And they can be developed for a variety of applications. But again, enzymes and pH are really half of this pie chart. And there are good reasons for that. There are some exquisite methods for changing a lanthanide chelate, having a change in water accessibility if the top of the basket where that holds the gadolinium is exposed, or change in ligand proximity, or tumbling time, or the whole T2 aggregation state could change. For CEST, the proton could change from exchanging slowly to exchanging fast, or vice versa. A change in electronic shielding can occur too. And so these are the six mechanisms. There are only really six mechanisms for responsive MRI contrast agents that are, the lanthanide chelate series, and then also for iron oxides and manganese as well. I'd like to point this out because there's a whole variety of agents that are responsive that have made much more progress in the literature looking at biomarkers of the tumor microenvironment. I'd like to switch over to discussing simultaneous PET MRI. This is a multimodal MRI session. Simultaneous PET MRI is an intriguing technology. We use it by putting an insert into a small animal MRI scanner so we can perform the PET and the MRI at the same time, truly simultaneous. So we can acquire FDG PET and DC MRI at the same time, as shown here. I'd like to pause and just focus on this yellow box. What is the advantage of simultaneous PET MRI relative to sequential PET and MRI? I'm asked that very often. And I really do not think it is the convenience or the cost, especially for small animal imaging. I can make a case in the clinic where time is money and so on. But really, I think where we're going to see the greatest benefit for the applications is when timing and spatial registration are important. And both the PET and the MRI have to be performed at the same time, or there has to be fantastic spatial resolution between the two modalities. Although this is somewhat mitigated by outstanding techniques for spatial registration that can be applied post-acquisition. So let me show you a couple of examples of that. Here's a case where we performed simultaneous PET MRI to measure FDG PET uptake, an indication of glucose metabolism, and then also a cytocest MRI, a method of measuring the pH of the tumor microenvironment, which is reflecting the lactic acid production from glycolysis. And in both cases, here you can see that the tumor pH actually went down, indicating more lactic acid buildup because of an inhibition of gluconeogenesis for this particular metformin drug. And meanwhile, the FDG signal decreased, showing that the tumor was negatively impacted by the therapy. But I also want to point out that this is an involved study. You have to time your FDG injection, your PET MRI preparation, position the mouse, your anatomical MRI, inject your MRI agent, do your MRI study. The PET is actually pretty easy at this stage. It's more like a radioactivity camera, if you will. But yeah, a lot of workflow goes into these studies, and that emphasizes that simultaneous PET MRI is certainly not easier to perform than sequential PET and MRI. And it takes the same number of personnel and attention to detail. Now, for the mouse, it's easier on the mouse or easier on the patient, just being scanned at 1 hour or one time, but certainly not easy to set up these studies. Here's an example, though, where simultaneous PET MRI can have a remarkable improvement. Here's an agent that's an MRI contrast agent with gadolinium that we've made in our laboratory, and a radioactive version radionuclide version with gallium-68. And we've co-mixed both of these agents together and put that into a phantom. We know the gadolinium to microcurie ratio. So we don't actually have to know the concentration of the gallium. We just have to know how much microcuries are coming from the gallium to get this ratio. Then we perform MRI and measure the R1, the T1 relaxation time constant with the agent. And then, of course, we have a control tube that has no agent, so we can calculate the delta R1 due to the contrast agent. We also detect the amount of activity, and based upon that gadolinium microcurie ratio that unfortunately I've hidden behind this ellipse right here, we can then convert from activity to gadolinium. That allows us then to compare delta R1 with gadolinium to calculate an R1 relaxivity. In this case, we got that number. And then using a predefined calibration curve for this agent, using samples placed in a phantom box that was well controlled at 37 degrees, and we fit this data with an equation. We can calculate what the pH is based upon the relaxivity, or yeah, and then that compares well with the benchtop MRI value for that particular sample. Now, if we have a high concentration of MRI contrast agent, this technique works quite well with a low standard error, and we get a nice linearity. Now, at lower concentration, MRI is a noisy technique, and so we don't have quite the accuracy or the precision that we would like from this methodology, but we are able to measure pH using the PET MRI system now. We have done that in some preliminary studies with mice. Here's a Miyapaka tumor where we've measured the delta R1 using MRI, and we've also measured the microcuries emanating from the tumor and then converting to gadolinium concentration. We can calculate R1. We did this over a series of 4 time points for this particular mouse, and after allowing the agent to accumulate in the tumor, of course. And then by taking the average, we can then compare with our calibration curve and read off the pH, the extracellular pH in the tumor microenvironment. Now, we don't have validation of this pH number in this particular case, but tumors are expected to be acidic, so it matched our expectation for this tumor. However, we did see some gallium dechelating from the agent, violating the assumption that the two agents have identical pharmacokinetics as they are traveling from the syringe into the tumor. And so we have made other variations that are more stable, and we're looking forward to testing those. But it does show an application where simultaneous PET MRI is required, so we can get the PET and the MRI at the same time in order to calculate pH. Finally, in the remaining time, I'd like to make a case for how molecular imaging can impact the clinical radiology operation. And this might seem intuitive at first, but it makes an important point. Here's pathology images, H&E stains of two types of breast cancer. On the left is a benign set of breast slides, and malignant breast cancer is shown on the right. And you can see that there do appear to be some different features, and if we were to train an observer to identify these features just in general, after a while your eyes would pick up on salient features relative to benign. You wouldn't even have to be instructed on what to look at. You could eventually train an observer to identify benign versus malignant within a training set. And of course, if you have more imaging features, higher magnification, then the training will be much faster. And that even at low magnification, in this case, the training set, after a while, an observer can identify benign versus malignant and then go to a new test set that the observer has never seen before and still get the right answer. So this shows that there's actual learning going on and isn't just memorization of the training data set when a new test set is shown. It's been shown using a flock sourcing model that four observers put together actually give a really good ROC curve. Let's look at another example of breast mammograms that are relatively featureless compared to pathology. And in this case, it's very challenging to identify the features between the benign and the malignant tumors. Training takes much longer, and unfortunately, there's poor learning that goes on. After a while, an observer can memorize the training set and identify benign versus malignant images. But then when the new images are presented to the observer, well, it's a coin flip. The observer really hasn't identified what features would distinguish between benign and malignant. And you can see they are quite featureless in that regard. So the take-home message is perhaps obvious to us, but if molecular imaging can provide more imaging features and turn standard radiology into something that's more like pathology, which is feature rich, well, that will shorten training times of our observers. It will improve learning relative to memorization. So we want a good learning experience here. This is a poor learning experience over here. And then improved sensitivity and specificity. And again, 0.99 ROC, fabulous sensitivity and specificity when four observers were combined. And that is key. We need four observers. Well, as you may know, it's challenging to get four radiologists together and look at each image. So how are we going to then solve that problem? Because after getting molecular imaging to turn standard radiology into a very feature-rich sets, we still will need four observers. Well, the key is that the study was not performed with human radiologists. It was performed with pigeons. Pigeons were placed in a box and trained to peck on an image when the pigeon identified a benign or malignant image. And all the data that I just showed you comes from this pigeon study. The observers were pigeons, which then begs the question, are the radiologists really afraid of AI and deep learning and data science, or are they going to be replaced by pigeons? Well, I'd like to suggest that we could implement radiologist equivalent pigeons for low cost, accurate cancer evaluations. We could make the replace program. I'd like to go with the Texas Pioneer Pigeon. This is a pioneering program in the state of Texas. It's really a handsome bird, if you ask me. And at $200, well, if we got four pigeons plus the maintenance with food and everything, well, it'd be about $1,000. And so if we calculate a 15-year lifespan, I could replace our 130 clinical radiologists at MD Anderson, saving about $840 million. So that's a sizable amount of money to save with a bunch of pigeons. So of course, I'm being a bit facetious here. I do want to go to work next week and actually have a job. So I'm not actually suggesting that we go ahead and replace all our clinical radiologists, but it is to show that molecular imaging can have an impact, where even pigeons can get the right answer if we provide enough features to our observers. And so I think that's an important goal for molecular imaging. Finally, I would just like to thank those in my research group who take crazy ideas and somehow get them to work. I really appreciate that. And I'd also like to thank our funding sources and our collaborators at MD Anderson and abroad. Thank you for your time.